David Helfgott is one of the rare cases of a very successful schizophrenic patient. He is now nearly 70 years old. He, when he was in his teens and early 20s, he suffered several schizoaffective episodes, which is a form of schizophrenia. He went on to move from his native Melbourne to, actually he lived in Perth for a while, to the Royal College of Music. But at about that time, he had several additional schizophrenic or schizoaffective episodes. He was then hospitalized in Australia for about 10 years. And from about 1984 to the present, he has rebuilt his concert pianist career. And according to the biography that his wife, who helped to save his career, wrote, this is the medication that he takes, chlorpromazine, which is a D2 receptor blocker, as well as an anticholinergic drug for the inevitable side effects of these blockers called tardive dyskinesia. Before we go on to discuss schizophrenia in more detail, let me remind you that we're having no quiz, and when word got around, attendance dropped significantly. You will get quiz-like credit for emailing Ralph Adolphs or myself a question for Friday's session. What is the challenge for neuroscience in the remainder of the 21st century? Give us, please, a reference from a journal. We're going to post we'll have sessions as discussion sections as usual tomorrow. The Friday on December 4th, the final exam will go up. It will cover the entire course, but will emphasize the second half, and it'll be due a week from then. You could turn it in early if you'd like. Schizophrenia is very much a cognitive disorder, and today's lecture combines really ideas of people whom I've invited to Caltech over the past decade or so to speak about schizophrenia. Eric Kandel from Columbia, who is the author of our textbook. Robert Friedman, a collaborator at the University of Colorado. These are all academic research psychiatrists. David Lewis, chair of psychiatry at the University of Pittsburgh, and Mike McIntosh, who's at the University of Utah. And we talked about Shine already. A movie that's a little bit more familiar to you probably is A Beautiful Mind, which is more recent. Does a pretty good job on schizophrenia. And toward the end of today's talk, if there's a chance, I'll show you a section from One Who Flew Over the Cuckoo's Nest. As opposed to major depression and bipolar disorder, we don't see much schizophrenia on the typical college campus. And that's typically because schizophrenics have a lot more trouble in a high-functioning environment than bipolar or major depressive people do. And also schizophrenia tends to come on just a bit earlier than the mood disorders. And so as a result, by the time people get to college, it's pretty evident that they are not functional. There are obvious exceptions. John Nash is, of course, a classic exception. As usual with schizophrenia, we go through the clinical description, the genetics, the pathophysiology, what we know about it, biomarkers and animal models, heterozygote advantage, there won't be much, in therapy. It's pretty clear that schizophrenia affects several complex systems in the brain. There are simulated interviews with schizophrenics on YouTube. I've got a videotaped interview with a real schizophrenic patient if you'd like. And it's pretty clear also, even though people have their first major break in their late teens, in retrospect, people say he was weird even as a child. There are issues of social isolation, withdrawal. there is impairment in normal function, and there is some odd behavior, some ideas. We would call these prodromal signs. Before a person becomes schizophrenic, there are plenty of people with these signs who are completely normal functioning. So schizophrenia has three major classes of symptoms. There are the positive symptoms, the ones that we all know about a schizophrenic patient, the ones that get schizophrenic patients into great trouble, the delusions, the hallucinations, the thought disorders. To give you an example of just one random example, you can think of people who've been in the news as mass murderers, etc., who have said that they suffered from delusions and hallucinations. We've been reading about the shooter in Colorado, and apparently a few years ago, when he moved into his present home, he told a neighbor, "You should have a metal roof because the government can listen through other kinds of roofs." Now, this is obviously not enough to diagnose this person as a schizophrenic, but one of the most common characteristics of schizophrenics is that they think that in ways that are appropriate to modern technology, people are listening to them. You may remember in A Beautiful Mind, the John Nash imagined, or Russell Crowe playing John Nash imagined, that he had a transmitter embedded in his arm. People think that the CIA is listening to transmitters in their teeth, or that they have receivers in their teeth, or that articles of clothing are broadcasting. And so the idea that people can listen in, can determine what they're doing, that typically higher organizations such as the government can do this, occurs very often in schizophrenics. Actually, though, a major problem with schizophrenia is also the so-called negative symptoms. Decreases in motivation, emotional expression. Generally, in some cases, you might, and I visited mental hospitals, typically in the VA system where there are lots of schizophrenics. People sit around, they are catatonic, they don't do much. And then there are the cognitive deficits. The cognitive deficits are the ones that are least amenable to modern psychiatric medication. People don't pay attention, they can't actually plan and execute, they have problems with some types of memory, and this is why we see very few schizophrenics in high functioning academic environments. Schizophrenia, Ralph gave you a wonderful lecture on autism, and he talked about Simon Baron-Cohen's ideas. And some people put schizophrenia on the other end of a diagnostic arrangement from autism. It's not clear why or how, but we're going to go back to the relationship between schizophrenia and autism in this lecture a bit more. So again, A Beautiful Mind is the classic delusion hallucination story. What is atypical about A Beautiful Mind is that in most cases, schizophrenics have their hallucinations and delusions in an auditory fashion. That is, they hear voices or they think that people are talking about them. In a movie, it's much easier to depict schizophrenia as visual hallucinations. And so throughout A Beautiful Mind, one sees these elaborate characteristics, these elaborate processes that are actually visual hallucinations. Let's talk about genetics. So it is very clear in almost every study that there is a family incidence of schizophrenia. Now we're going to spend more time talking about genetics for schizophrenia than we did for bipolar disease and that Ralph did for autism. And this is because of some wonderful advances that have occurred in the last 2 years. The general population has between a 1 and a 2% chance of diagnosis of schizophrenia. And this is remarkably independent of the culture or of the society. So, on the other hand, the concordance between identical twins, that is, between two people who share 100% of their DNA, approaches 0.5. Among studies, it could be as low as 0.4, it could be as high as 0.8 among studies. In general, people say it's roughly 0.5. And as the amount of shared DNA increases, the concordance decreases. So for fraternal twins, not identical twins, it drops to 17%. So people who share 50% of their DNA have a still much larger concordance than the general population, but much less than identical twins. How it is and what the mechanism that 50% sharing has such a great range, we don't know. Then when we go to 25% for half siblings, grandchildren, etc., the concordance is less and significantly different from the general population for first cousins, but less again. So what do we say about the general characteristics of genetics of schizophrenia and of complex diseases in general? First of all, that the disease is polygenic. It occurs only if several genotypes are present together. There is no one disease that causes schizophrenia. There need to be several together. In addition, the disease is partially penetrant. So here we have a concordance not of 100%, but of 50%. So there clearly are non-genetic factors, epigenetic factors, environmental factors, or the disease could be inherently stochastic, whatever that means. I'll come back to environment a bit more toward the end of the talk. And finally, the disease is genetically multifactorial. That is, if a set of 2 or 3 or 8 or 10 genes needs to be malfunctioning or needs to be mutated in one population, then another set might be mutant in another population. And it is this multifactoriality that bedeviled complex disease researchers for many years until, as you'll see in the last 2 years or so, when people were finally able to get the throughput of genetics fast enough and detailed enough so that they could overcome the genetically multifactorial problem. But these three characteristics together cause major problems for assessing schizophrenia and other complex neuronal diseases. Any questions? So in a sense, if we go back to DSM, which Ralph has done and I have done, and we ask, what is the relationship between clinical neuroscience and molecular neuroscience? Clinical neuroscience is this big, thick book which psychiatrists use when they testify in court, when they diagnose patients. Molecular neuroscience is the human genome, animal models, associated data. What's the relationship between them? One, probably the key element here is the so-called SNP map of the human genome. So all 3 billion or so base pairs are not identical among us. And there are changes along the way. There are something like these days 3 million single nucleotide polymorphisms at a frequency of about every kilobase, although the frequency varies a great deal among the genome. Most of these are in non-coding regions. Even if they are in coding regions, even if they lie within coding regions, they may be synonymous. They may not encode a different amino acid. And even if they do encode a different amino acid, there may be no functional significance, but they're like signposts. They give a way for a person to figure out what is different between two genomes. haplotype, which we won't discuss much, is actually a pattern of several nearby single nucleotide polymorphisms. So, for instance, here there are two SNPs, here and here. And so you would think that 2 to the fourth, 2 squared, would give 4 possible haplotypes, but actually only 3 of them exist. And so some of these haplotypes are correlated with each other. We won't discuss them much because this is not a course in genetics. So how do we hunt for genes in SNPs? Well, here we have some controls, and here we have some schizophrenics. Let's look at locus A, which might be on chromosome 12. We have 2 sequences, and it turns out that controls, 20% of controls have this sequence, 80% of schizophrenics. In controls, 20% have this sequence at the locus, 80% have another sequence at the locus, A1 and A2. The same is true for schizophrenics. So there's no linkage to schizophrenia. However, at locus B, which might be on another chromosome, for instance, chromosome 8, as we'll see later in the talk, chromosome 6 is very significant these days. Actually, controls have 40% of sequence B1, schizophrenic, and 60% of sequence B2. But schizophrenics have a different ratio of B1 and B2. And so that tells us that locus B, but not locus A, may be near a gene that helps to cause schizophrenia. Not that causes schizophrenia, not that this particular difference in sequence causes schizophrenia, but it might be near a gene that helps to cause schizophrenia. So you can say the same thing by looking at the difference between controls and schizophrenics, and the same conclusion comes true. And maybe near a gene means that recombination is so infrequent that it has not occurred. So near a gene means that they have remained together during some aspect of human history. Of course, chromosomes stay together. Chromosomes do get shuffled because of recombination, and if they get shuffled only partially, then 2 sequences stay near each other. Within the past 10 years, it's become evident in some cases that large deletions of chromosomes seem to be associated with schizophrenia. So this group, large, well-funded, and hardworking group, said that common genetic variants don't seem to have a major impact on the polymorphisms, on the predisposition for schizophrenia. But this group did find very rare, very large deletions and duplications, hundreds of kilobases, megabase, may be associated with a minority of schizophrenia cases. The paper said we can't focus on a finite set of pathways. One event was near a gene that interacts with one known mutation associated with schizophrenia in one family. But this is pretty slim pickings. Clearly, schizophrenic genetics, now they say, must turn to the identification of rare contributors. However, in this study, which looked at large copy number variations, large deletions, the authors suggested that we really do need to do whole genome sequencing, complete genome of patients who have been diagnosed by psychiatrists. So that's the way matters stood. Oh yeah, CNVs are copy number variations. I apologize. So that's the way matters stood until a little bit, about a year and a half ago, when a paper written by 208 authors appeared. And this paper has Uh, the 208 authors are the Schizophrenia Working Group of the Psychiatric Genomics Consortium. So there is around the world a loosely organized group of people who try to do the genetics of psychiatric disease. And they set up, this is a very democratic, very loose organization that sets up individual disease-oriented working groups. So there is a working group on depression that has produced very little so far. As I mentioned on Monday, there was a disease working on bipolar disease. Sorry, there was a group working on bipolar disease. And most experts thought, because of the apparent simplicity of bipolar disease, because of the ease of diagnosing it, and because of its very high heritability, that bipolar disorder would be mapped before schizophrenia. That has not turned out to be true. In fact, schizophrenia has been mapped. This is called a Manhattan plot for obvious reasons, that most significant genes tower above the others. It is a plot of the significance of a polymorphism and its association with the disease in question, here schizophrenia. So these are workers. This is a summary of data from 37,000 cases in 113,000 controls all over the world, in which people tried to standardize the diagnosis and, of course, standardize the molecular genetics. Down here at the bottom, we see chromosomes. As you know, the longest chromosome is number 1. The shortest is 22. There's also the X chromosome. And so here is the usual standard for significance above 10 to the minus 8.5. So everything above this red line is a SNP, a polymorphism that is significantly associated with schizophrenia in this comparison of 37,000 cases and 113,000 controls. So 150,000 patients, probably a million or so PCRs for each of those patients. Lots and lots of data. Enormous paper, lots of software, lots of analyses, which are still ongoing. This one stands out, and yet we don't know what it means. We know that it's on chromosome 6, but the annotation in the database says that and there are about 8 open reading frames associated with genes in them. Nobody has ever identified one of these genes before as related to schizophrenia, and I can't tell you a story about it, except that it's obvious. Here's one that I can tell you a little story about. It is on chromosome 12. The human name for the gene is this. It is a voltage-gated not a California channel, a calcium channel. It is, in fact, the alpha 1C subunit of a voltage-gated calcium channel. It clearly, some mutations in this gene are also associated with an autism spectrum disorder called Timothy syndrome. And you'll remember I told you in the last talk that the modern trend at the NIMH is not to do research on the basis of diagnosis, but to do research on the basis of neurobiological phenomena that exist across diseases. And so one of those neurobiological phenomena obviously is synaptic transmission. And so this gene, calcium channel subunit, is quite interesting. There are others. In fact, if we look at the 108 identified genes that came out of the GWAS study that I just told you, 83 of them were not previously reported. Happily, many of them are expressed in the brain, most of them. And everybody was relieved to find that a gene which came out prominently was the dopamine D2 receptor, which is a G protein coupled receptor. And I will tell you about the D2 receptor in a few minutes. Another GPCR, the metabotropic glutamate receptor number 3, has been identified as a candidate for schizophrenia therapeutics, but has not yet been successful. Also, some genes were identified in the immune system. The immune system has been identified with schizophrenia possibilities before. I'll tell you a little bit about this. Glutamatergic transmission, well, that is an easy one because most synapses in the brain are glutamatergic. Other genes associated with synaptic function and plasticity. I told you about the calcium channels, and there are several other neuronal ion channels also came up in the GWAS, and several genes associated with neuronal development. And as I've mentioned before, even though we don't think that autism and schizophrenia are the same phenomenon, there's a partial overlap in some cases with genes identified in autism. I just gave you one, a calcium channel, and in other intellectual disabilities. One gene that continues to come up and that came up in our lecture on development of the neuromuscular junction, one class of proteins, we called it then acetylcholine inducing activity, and it is now known to be a member of the neuregulin family. Neuregulins are transmembrane proteins, and in some cases, they are proteolyzed to release a growth factor with an epithelial growth factor-like domains. The actual topology of these proteins differs on whether they are type 1 or type 3 or type 4, but in most cases, the proteolysis releases a fragment. Now, what this does, we're not sure, but this is a gene that comes up over and over again in psychiatric disease, in neurodevelopment. And it's one example of a gene that we like to tell stories about in this field. All right, so now we come up with the challenge of relating genetics to biology. Actually, it's a much older challenge than that, because in fact, schizophrenia has been a challenge for neuroscience for about a century. Early in the 20th century, the Germans, who loved to classify things at the time, started classifying psychiatric diseases. And unfortunately, this was helped by Nazis, who were among the people who said that the way to get rid of people in society is to classify them as being psychiatrically deranged. And so the Nazis, as part of their eugenics, decided that most of schizophrenia was genetic. And as a reaction to that, American psychiatrists in the 50s went in completely the opposite direction, and they came up with the refrigerator mother, or the schizogenic mother hypothesis, in which all of schizophrenia, possibly all of autism, was based on upbringing and none on genetics. Bettelheim has been pretty much discredited as an authoritarian figure and also for fudging his data. But this is a typical transition in the schizophrenia field. First, genetics governs everything, then no genetics at all. It's all environment. Since then, almost every advance in biology has been tried out, thrown up against schizophrenia to see whether it sticks. A story that's told around Caltech is that Linus Pauling, in the early 50s, fresh from his discovery of sickle cell anemia, fresh from his discovery that a disease could have a molecular basis, said, oh, maybe all diseases have a molecular basis. Maybe even the toughest diseases have a molecular basis. maybe schizophrenia. And so he took the most advanced chemical technique available at the time, and he applied it to the urine of schizophrenic patients. That most advanced chemical technique was paper chromatography. And you've done this in high school, right? You cut a piece of filter paper or a paper towel, you put it in an inkwell, you get a bunch of spots, and you say, well, clearly the ink has several chemical components, and that's what Linus Pauling did to the urine of schizophrenic patients. Uh, he didn't find anything, and in fact, there were confusions with medication, etc. But he came up with a grand term called orthomolecular psychiatry, and he published an entire book on orthomolecular psychiatry. So the hope that you could take a scientific advance and apply it to schizophrenia persisted. In fact, in 1955, when chlorpromazine was discovered, um, this chlorpromazine was originally identified as an antihypertensive drug, a drug for hypertension, but it was pretty quickly realized to be a dopamine D2 receptor blocker. And so that gave rise to the theories of dopamine uh, as as an important transmitter in schizophrenia. So this is a plot of the number of patients in U.S. public health mental institutions as a function of time since the early 1800s. Remember, schizophrenia is very common. Well, it's at the 1% level in every society. And so a lot of schizophrenics in the US population were in mental hospitals until the mid 50s, when attitudes changed partially as a result of the introduction of chlorpromazine. And so people were released from mental hospitals and unfortunately did not get the kind of support that they needed. So great advance scientifically, chlorpromazine led to dopamine. A few years later, people realized that glutamate was the major transmitter in the nervous system. Actually, it was fairly late that uh, this became obvious, and so people started wondering about glutamate as a figure in the pathophysiology of schizophrenia. One characteristic of all of the theories, of all of the possibilities, is that they don't go away, they continue on in research on schizophrenia. So glutamate is still thought to be at fault in schizophrenia, and there are drugs that target glutamate receptors. We began learning about developmental neuroscience in the 90s, and so people started understanding that there were prodromal effects of schizophrenia, and so there were ideas about development and migration of neurons that are still being researched in schizophrenia. Beginning in the year 2000, we began having the power to do genetics and genomics, and so people started hunting for schizophrenia genes. Actually, Linus Pauling had talked about a schizophrenia gene several decades before that. He didn't have the concept, actually, of polygenic and multifactorial diseases. He thought there might be just one gene. We continue to do that. Several years later, people began understanding that there were enormous diversity among GABAergic interneurons and began wondering whether one or another class of GABAergic interneurons was at fault or could be the cause of schizophrenia. That goes on. Beginning about 10 years ago, when we started learning about inflammation and more about immunology, people began thinking about inflammation as the cause of schizophrenia. So there's no satisfactory explanation yet. When you become professional neuroscientists, may tackle this in a better way. But in general, people think about schizophrenia as they do about depression and autism and bipolar disease as abnormal balance among circuits rather than individual neurons that either degenerate or fire too much or too little. Neurodegenerative disease, clearly neurons die. Schizophrenia, they do not. They don't function well, but they don't die. There are nonetheless gross and neuroanatomical abnormalities. Here's a figure from your text in which the cerebral ventricles are increased in size in two people who are discordant for schizophrenia. This one has schizophrenia, this one does not. This does not mean actually that this is caused by abnormal ventricular size. It means that the rest of the brain is smaller, allowing room for the cortex. A common observation is decreased gray matter, that is, the cell bodies and the dendrites. Not so much in white matter, but there is decreased gray matter in schizophrenia. And always the question arises, well, is this a result of medication? Because there are very few non-medicated schizophrenic patients around. Apparently it is present in first order, never medicated patients as well. So here's an example from some figures about decreased numbers, modestly decreased numbers of neurons in hippocampus. I told you there's no decrease. In some cases, there are modestly decreased numbers, but mostly the complexity of the neurons is decreased in schizophrenia. Here's an unaffected twin with an unaffected person with lots of spines. Here is two schizophrenic patients at autopsy with many fewer spines. And so the idea has arisen over and over again that neurons become less complex, they don't function very well. Now, another interesting point is that the hallucinations and the delusions actually can be measured in an objective sense, so that some neuronal activity actually occurs in primary cortex in hallucinations. Auditory cortex, which, as I told you, is the place where lots of delusions occur, has activity when a patient says, I'm having a hallucination. So I've told you something about genetics, something about pathophysiology. One has to remember that the concordance among identical twins for schizophrenia is only 50%. So the question becomes, what causes the variability? What causes the other 50%? Well, there is a fair amount of evidence that the fetus's nourishment and health is involved in the development of schizophrenia. The late Professor Paul Patterson at Caltech, who died about a year and a half ago, was one of the pioneers of this field. He discussed viral infections during pregnancy. Some of this work is continuing among his former students. The idea is that these viral infections possibly led to low-level inflammation, which has a significant and continuing influence on the function of the brain. Another point that other people bring up is head injury. So my collaborator, Bob Friedman at the University of Colorado, has taken me on initial interviews of schizophrenic patients. And the first question he asks is, have you been in a car accident, fallen off a horse, or had another kind of brain injury recently? It turns out that people who have a predilection to, who are at risk of schizophrenia may have the final insult to be a head injury, and that then precipitates schizophrenic episodes. So these are a couple of the non-genetic characteristics. What about biomarkers, as Ralph called them? Objective physiological measurements that correlate with schizophrenia. Biochemical, physiological. Instead of having a psychiatrist sit down and ask a patient, tell me about your delusions, are there any objective measurements we can make? Well, this is going to be very tricky. Here are statements from NIMH, but biomarkers would be really helpful. Maybe biomarkers could aid people in subcategorizing schizophrenia so that a disorder could be broken down. This was apparently not necessary in the recent GWAS, Genome-Wide Association study, but it would still be very helpful. So two biomarkers that have proven helpful have been electroencephalograms and eye pursuit. Now, Ralph gave you a wonderful set of examples of eye pursuit, of eye movements, when he was discussing autism. I'm going to talk about electroencephalograms instead. So here, these are actually data from my lab on a phenomenon called sensory gating. And what one does, this is a mouse model, and it's not schizophrenia, but what one does is to put the electrodes on specific regions of the cortex to measure evoked potentials. And typically one can do this with 2 clicks. So here is the click. The electroencephalogram always shows a series of peaks after a click, but after the second click, typically in most subjects, a couple of the peaks are smaller. This sensory gating, it's called, this decrease in P50, 50 milliseconds after the click, is found to be defective. That is, the suppression does not occur in most schizophrenics. But I should point out also that this anomaly in sensory gating, in which the second peak is not much smaller than the first, also occurs in 8% of the population, which is 8 times as many people as there are schizophrenics. And so if we come to my office hours in the afternoon, and we did a sensory gating experiment on 100 students or faculty members who walked by, we'd probably find normal sensory gating, this pattern, in 90 of them, anomalous sensory gating in 8 or 10 of them, but maybe only one of them would be schizophrenic, if that many. And so there's very little predictive value in this, but there are genetics that suggest that the sensory gating abnormality has autosomal dominant transmission. Dominant, you understand, autosomal means it's not a sex chromosome, but one of the other chromosomes. And one sees this sensory gating anomaly even in healthy relatives of schizophrenics, meaning that it's not a consequence of medication. And so, sure enough, this trait maps near the alpha-7 nicotinic receptor, which we will discuss a little bit later. So there are getting to be some biomarkers, smooth eye pursuit, the way a person tracks, some electroencephalographic anomalies, but they're still pretty crude and not predictive. So now the question becomes, is there an advantage of carrying a few genes for schizophrenia, a few of the 108? Does this confer an advantage to a person in his social life, in his work life? None is known. Again, schizophrenia may be inherited. An example that I mentioned earlier in the lecture, if you've seen the movie Shine, you know that David Helfgott's father was a mean person, and he could have been the gene carrier for the schizophrenia. Another example is that John Nash's son is a schizophrenic mathematician. And so there's clear genetics, as I've showed you over and over again, but there doesn't seem to be any heterozygote advantage. Now, we have to contrast this with cystic fibrosis. We also contrast this with bipolar disease. In cystic fibrosis, the most common inherited disease in our society, the heterozygote carrying only one allele for the mutation is resistant to dehydrating diseases. And this may be important in societies where cholera is widespread, but there doesn't seem to be any in schizophrenia. And again, we contrast this with bipolar disorder. Next question is therapeutic approaches. Next point is therapeutic approaches. I mentioned chlorpromazine, which was really the first medication found. There are two classes of antipsychotic, anti-schizophrenic drugs. There are the classical or typical ones, and then I will tell you about atypical ones later on. This is a graph that dates from the 60s. On the x-axis is the drug potency, Ki, how tightly a drug binds to a receptor, the D2 receptor. And on the y-axis is the average clinical daily use that a clinician would use to suppress the symptoms of schizophrenia. This fairly good correlation indicates that the more tightly a drug binds to the dopamine D2 receptor, the better it is at suppressing the symptoms of schizophrenia. Now, it takes a couple of weeks, so there's this therapeutic lag in schizophrenia that I discussed in great detail for antidepressants. The therapeutic lag is a little less in schizophrenia. 2 weeks is typically enough to tell whether the drug is going to work or not, but the full-blown effect of a drug is 4 to 6 weeks. Given that therapeutic lag, nonetheless, there is this interesting correlation between a drug's potency on dopamine D2 receptors and its effect on schizophrenia. This potency is not a good correlation if one looks at another GPCR, the serotonin 5-HT2A receptor, or for alpha-1 adrenergic receptors, or for histamine H1 receptors. These correlations do vanish. And so for classical antipsychotic drugs, we think about D2 receptors, but we don't know why. In fact, there are lots of effects of GI-coupled receptors. The dopamine D2 receptor is a classic GI-coupled receptor. I'll just remind you of some of those effects. One of them is activation of GERCs, G protein activated inward rectifier channels. Another effect of activating GI-coupled receptors is to decrease cyclic AMP. We've discussed that in the context of electrophysiology in the hippocampus. And in general, then, one changes gene activation as well with GI-coupled receptors. So how binding to a GI-coupled receptor and the effects on the GI gene, the GI protein decreases hallucinations after 2 or 3 weeks is not at all known. In fact, I showed you a quote from Hyman, Nestler, and Malenka last time. How do psychiatric drugs work? We talked about antidepressants, and they say very much the same thing about antipsychotic drugs. Their full therapeutic actions take weeks, suggesting that slowly developing adaptations, in this case to the initial D2 dopamine receptor blockade, are causing the antipsychotic effects, but nobody knows how. To make things even more complicated, the current generation of antipsychotic drugs, the so-called atypical antipsychotic drugs, work more on 5-HT2 receptors than they do on D2 receptors. And so we don't understand at all. But maybe you will understand how drugs suppress schizophrenia. In fact, there is another problem with schizophrenia, and this is the 3 classes of symptoms: the positive, the negative, and the cognitive. None of the drugs touch the cognitive effects. And so Robert Friedman has been emphasizing the use of cholinergic drugs to act on the cognitive effects of schizophrenia. And so remember I told you about the nicotinic alpha-7 receptor. Friedman would like to say that the cognitive effects occur because the brain is all mixed up, and that acetylcholine and GABA help to filter the signal from the noise. And so let's look a little bit more at the alpha-7 drugs, the nicotinic drugs. As I told you, the sensory gating anomaly maps near the alpha-7 nicotinic receptor. The recent GWAS studies did not find a hit here, but actually this region of chromosome 15 is one of the worst to study in the human genome because it has lots of duplications and omissions. And so the fact that you can't do a good SNP map near it probably is uninformative. So several companies are testing alpha-7 agonists and modulators for cognitive enhancement. One set of genes that did show up in the GWAS study is a cluster of nicotinic receptors, the alpha-5, alpha-3, and beta-4 cluster, also on chromosome 15. But why is this all significant? It's significant because 90% of schizophrenics smoke. And schizophrenics smoke very differently from the usual nicotine addict. The usual nicotine addict keeps his fingers over the filter, carefully adjusts the amount of nicotine that he gets in. Schizophrenics tightly put their fingers over the filter, take long drags, deep drags, and keep the nicotine in the lungs for a long time, as though they were trying to get much higher nicotine concentrations than the typical smoker. And indeed, the alpha-7 receptor is less sensitive to nicotine than other nicotinic receptors. And so the theory has arisen that schizophrenics are trying to activate their alpha-7 receptors with this different mode of smoking. And most movies that you say with schizophrenics, they have cigarettes in their mouth all the time. Russell Crowe in A Beautiful Mind, Jeffrey Rush playing David Helfgott in Shine, for example. So speaking of cigarettes and smoking in schizophrenia, I can show you a clip from One Flew Over the Cuckoo's Nest. The original novel was by Ken Kesey. The movie, 1975, starring Jack Nicholson, got an Academy Award. Also, Louise Fletcher playing Nurse Ratched, Best Actress. It's time to go, but if you'd like to stay, I will show those of you who want to stay a clip in which Sidney Lassick wants his cigarettes so badly that a riot scene occurs. If not, see you on Friday, and don't forget to send us ideas for Friday's discussion. Okay, I will play this. Gotta move it.
Cigarettes, please, Miss Ratched. Forget the cigarettes, Cheswick. Cigarettes are not important. Sit down, will you? For Christ's sake. Cigarettes. Miss Ratched.
Mr. Cheswick, you sit down.
Yes, but I'm going to go.
Sit down, Mr. Cheswick.
I want to. Christ's sake. Cigarettes.
Can everybody hear this?
Miss Ratched.
Mr. Cheswick, you sit down.
Yes, but I'm going to go.
Sit down, Mr. Cheswick.
I want to. Give him a cigarette, will you, Harding?
The last one.
It's a fucking lie. Why don't you give him a cigarette?
But look, I'm not going to shout you, Wilson. Come on.
Look, I don't want his cigarettes, but I want his, or 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 his yours. Do you understand that? I want my cigarettes, Miss Ratchet. I want my cigarettes. I want mine, Miss Ratchet. What gives you the damn right to keep our cigarettes piled up on your desk and to squeeze out a pack?
I forgot to tell you that all this takes place in a mental hospital.
What you feel like it, huh? Mrs. Ransom.
Mr. Harding,
I'm sorry.
You surprised me.
Not that I lost my head, I'm sorry. I'm really, really sorry. I just forgot. I'm really, really sorry. I just forgot.
It's all right. Miss Ransom.
yes, Mr. Cheswick,
I asked you a question.
I heard your question, Mr. Cheswick. I will answer your question as soon as you've calmed down. Are you calm, Mr. Cheswick?
Calm.
Good. Sit down, gentlemen. Sit down, sit down.
This on your fucking balls. Cheswick, I want you to know something right here now, Miss Ransom. I ain't no little kid.
You sit
I ain't no little kid. When you're gonna have cigarettes come from me like cookies, and I want something done. Take that right there.
That's right, now will you sit down?
I want something done. Something done. I want something done. I want something done. I want something done.
Okay, so the lesson is that schizophrenics really want their cigarettes. And one point that one might make is whether you could do some good in cognitive treatment of schizophrenia with vaping, which has very few of the side effects of smoking. I've actually seen very few discussions of this point.